so specifically recently, the interaction between metabolism and neural activity across the entire Drosophila brain. And I'm just going to share with you like the most recent work that I've done in collaboration with Stéphane Denis and Surya Ganguly, who are both also at Stanford. So this work kind of comes down to a big fundamental question, which is sort of how signals are coordinated across the brain. And signals can be sort of, we typically think of them as being neural activity, but it can be more than just that. And so like, just a quick reminder that, there's a bunch of different ways that we do this. And, I've been trying to pull, I'm old now, at least for a postdoc. So I've been trying to pull from the vast hoard of experience I've had with like other model organisms and other analysis types to try and make a coherent story that works in Drosophila. And of course, when we think about how signals are coordinated, we like oftentimes there's, for example, like here, the example of stimulating and recording. So in this case, this is not a fruit fly. Being optically stimulated and while neurons are being recorded from in a different part of the brain. Similarly, you can look at this in a whole brain setting. Again, also not a fruit fly here, but you can do, so people are performing fMRI type whole brain imaging, or even like whole cortical or mostly cortical calcium imaging while stimulating, or even also recording from neurons as well. You can also use multiple different sensors. So this is actually from a fruit fly from Tom's lab, where here you're, a former graduate student compared, in this case, voltage and calcium, which are two essentially neural activity proxies. So, and then of course, the goal here is to do multi-signal whole brain analysis. And really the pioneers for this, you have to look to human neuroscience. And this is just an example of combination PET and fMRI imaging done in a human, where in PET you can look at different molecule uptakes. So in the case that interests me the most, FDG PET, where you're looking at glucose uptake as it compares to neural activity as measured through BOLD. But I'm going to here to tell you that actually the fruit fly is probably the best model organism for looking at this kind of multi-signal whole brain imaging, at least currently. And we started with the question: can we use our relatively fast sensors that have high signal to noise to measure interactions across the whole brain? And I don't want to spoil too much, but the answer is we can. So Drosophila, for those of you who aren't familiar with it as a tool for neuroscience, is actually quite ideal for this kind of work. So we can do horribly unspeakable things to a fruit fly and therefore gain optical access to the entire brain. Here I'm showing just two examples, an anterior type dissection where the face is essentially you're looking through the face of the fly, or what a lot of people do, which is a so-called posterior dissection, where you're looking at the back of the fly and the fly is able to behave or see stuff and isn't in a weird kind of like body horror movie. And we also have access to a huge number of sensors. Like if you can think of a sensor, there's either it either already exists in Drosophila or people are going to build it. And I'm going to be talking about 4 specific sensors for this talk. At the top here, your classic GCamp, which is, again, a proxy for neural activity as it increases fluorescence to in response to increases in calcium. So when a neuron, a neuron fires and calcium is released, essentially this is your proxy for that. And also a red-shifted version of this, the R-Gecko family of indicators. It operates essentially the same way that GCamp does, but it's a red-shifted protein. And then also 2 sensors for or proxies for metabolism: the IATP sniffer, which binds ATP and increases in, fluorescence, and also the pyronic sensor, which does the same to pyruvate. So we have access to a lot of optical sensors, and of course, the most important thing for whole brain imaging is reliability and across animals. So in this case, I'm going to be using atlas alignment tools, that is to put every single brain into common brain space and use a commonly defined atlas, so that when I say, a certain brain region, everyone else knows what that brain region is, and they can have the same atlas. And again, this is stuff that is more or less cribbed from human neuroscience, but is a thing that fruit fly scientists now have access to, and it's great. So I'm going to talk today about 4 basic things. Quickly, I'm going to show you how we use the established methods for whole brain imaging to investigate metabolic networks, and that is whole brain correspondence of metabolic signals. And then I'm going to use the same method to compare signals between neural activity and metabolic flux in each individual region across the entire brain. And then I'm going to look at necessity and sufficiency of neural activity to drive metabolic flux and also to structure metabolic networks in the fly brain. And then finally, I'm going to end with just like the use of a simple behavior to investigate how the coordination of neural activity and metabolic flux occur just during sort of normal neural activity. So let's get started with this. So previous work that I've done in collaboration with postdoc at UCSF developed this pipeline, this analysis pipeline, essentially where a whole animal is mounted in a collar, and I have, again, like I said, optical access to the whole central brain. And I'm going to image the brain for both GCamp and also for a structure, what I'll call a structural image here. We don't need to go into the details of that too hard, but basically this allows us to align each individual brain to a template, use that template, which is to align our atlas, and from each brain region here, extract a time series based on that region. So, and hopefully this makes sense. But then what we do after that, in order to build a functional network, we basically take our different regions of interest and we take a time series from each one. So here I just grabbed a couple example bits here where you can see that we'll build a matrix by comparing every single region to every single other region. And as you can imagine, this gets rather large with the more regions you have to analyze. And then what I'm going to do is that for each ROI comparison here, I'm going to do a time series correlation. And that just looks sort of like something like this. If you plot this where the data in one region is compared to the data, the time series data in another region, and we call them functionally connected if there's a reasonable correlation between these two regions. And so this ends up being like what the sort of ground truth functional network of the brain, of the Drosophila brain that we first derived a few years ago, again, with Courtney Gallen at UCSF. And, I'm not going to spend a lot of time talking about the specifics of each region here. So don't get too weirded out by the fact that there's a big matrix here with a bunch of things that you maybe have never seen before. The only point here is that there is structure to this and that some regions correlate, more than others, right? And so this is what we call a functional network. And you can visualize something like this here in this hot mess of different brain regions, showing weights between different regions and how they interact. So with that, our goal, knowing this, was to take a look and see if we could, if there were observable metabolic networks. So in this obviously crude version of energy metabolism in a cell, pyruvate essentially is the byproduct of glycolysis. Eventually becomes part of the citric acid cycle, and of course, again, oversimplified. Eventually, we end up producing ATP, which is a sort of the main molecule of energy here. And then we're kind of interested in looking at how these interact with neural activity. And so we have the pyronic sensor here to measure pyruvate concentrations. We have the IATP sniffer to measure ATP concentration, and then we're going to use GCamp and JR-Gecko, like I mentioned earlier, to measure calcium as our proxy for neural activity and see if and take a look at, I guess, again, using our whole brain network style analysis. So in repeating my previous work, with a slightly different GCamp sensor, We can essentially reproduce, again, don't get too bogged down by this other than the fact that there's structure to this. We can see that there is, a structured network here. And then if I look at the pyronic sensor, again, using the same kind of method, just measuring pyronic instead, you can see that there is similarities here, hopefully, that there's, maybe visible this diagonal here, this little box here, some other features. Again, well, I don't want to dwell on it too long. And then also the same is true when we just look at the IATP sniffer, the sensor. And this is kind of initially interesting because a priori, there's not necessarily any reason to suspect that there are metabolic networks, right? You wouldn't necessarily assume that, right? So this means that when we just look at pyruvate concentration or ATP concentration, that there is structure to that across the brain when there doesn't necessarily need to be. And furthermore, as I'm going to show at the bottom of this, that it corresponds very well with the network that we observe with calcium. So if we just sort of take the upper triangle of these matrices and compare them as if they were sort of a linear time series like I was doing initially to build these. Again, this is going to be, there's a lot of correlations in this talk, so I hope it doesn't get too in the weeds about it, but this is now going to be comparing a correlation matrix using a correlation. So if we compare the matrices of pyronic and GCamp, there is good correspondence. So the R value here is nearly 0.7. That basically just means that every single point here corresponds to a single dot here and compares to the single corresponding dot here. Additionally, like with our ATP sensor, unsurprisingly, based off just eyeballing these things, it also corresponds well with the GCamp signal, and if anything, actually a little bit better than pyronic does. And that, of course, maybe unsurprisingly as well, again, that the two metabolic sensors correspond with each other very well. So that first part here, we've now established that metabolic networks exist, and that not only do they exist, they share common structure with the calcium networks. There could be some fun maybe talk on the Q&A about sort of what the thoughts are behind this, but we got excited. You always wonder what the order of experiments that were done for a talk or a paper, but this actually was the first thing that we did. Just to kind of test the waters for this. And so when we found such good correspondence, we knew that there was something very interesting here. And the idea was now to see what causes it and how can we describe it. So the next step was to compare these signals across the whole brain in each brain region. And basically, like I said here, determine the correspondence between the two. So in order to do this, I'm switching to the red-shifted calcium indicator, our gecko. And I'm going to combine that with either the ATP sniffer or pyronic, and I'm going to simultaneously image them in the same animal. And if you just take a look at a time series trace of a few of these things, again, this is just an example to show blue is always going to be gecko or calcium, green is always going to be the ATP sniffer, and orange is always going to be pyronic. So that'll stay true throughout the talk in case I left out a legend at some point. So you can probably notice that there is somewhat higher frequency activity that's observable in the gecko signal here, but that actually, if you look over the time course, which again here is several minutes, that actually the signals correspond fairly well. And if we break this down and look at individual examples of it here, I'm going to pull from this region here again. Don't worry too much if you're not familiar with what these are. These are just example regions, and I'm not going to go into it too hard about what regions do what, so we can just kind of ignore it. But if we take a look at the saddle here, I'm going to compare now the two time series between these two and see that there is good correspondence between them. But if we low pass these, if we only look at the lower frequency data that is below 0.1 hertz, the correlation between the two is actually, if anything, better. And that if we look at the higher frequency components of these, that actually the correlation between the two drops to near zero. In this case, it goes to zero, but obviously I cherry picked one to make it look more profound. But if we compare all ROIs across all flies with both sensors, or all three sensors, that is, you can see that the correlation between the two with the ATP sensor peaks at about 0.3. So it's like good. And then it drops off precipitously, essentially, to not to zero, but to low at higher frequencies here that we measure. And the same is true for the pyronic sensor here. The correlation is never quite as good as with the ATP sensor, but we see a similar drop off at higher frequencies. So really, the correspondence between these two happens at in low frequency space, not 100% exclusively, but it's definitely dominated by low frequency signal here. So with this, we clearly can see that when we image, when we do dual imaging experiments, that metabolic flux itself correlates well with calcium flux, and this is true across all brain regions. Oh, and I probably should have mentioned, actually, I probably should have been clear here that it is region specific, that when you shuffle the regions themselves, that there is essentially no correlation. So it's not, it could have been that there was there could have been brain-wide signals that correlated between the two, which is certainly easily imaginable. But actually it appears that this is just a region-specific event, which again further suggests that neural activity is somehow responsible for this. At least to me it did. So to get further into the weeds on that one, we wanted to test whether neural activity itself was necessary for metabolic network structure and correlation between the two signals, and also whether or not neural activity is sufficient to drive metabolic flux itself. So to test this, I did what I suppose could be considered a rather crude experiment, but effective. And that was to do the same preparation, same whole brain imaging method, and look at signal before and after the application, whole brain bath application of tetrodotoxin. Tetrodotoxin blocks essentially neural activity for the most part, essentially blocks action potentials. And hopefully, as you can see here, that in our gecko signal, our pyronic signal, and in our ATP sniffer signal, that there is essentially a reduction in the overall variance, like essentially a flatline of these signals in these corresponding regions. And if we look at this more, scientifically, it's very clear that the overall variance of your signal is quite reduced in all three of these sensors after you've applied tetrodotoxin. So like the unity line is here, and so significantly reduced, although not zero, signal across the brain regions. And if we look at this in frequency space, the relative power of the signals also drastically reduce. It's much more profound with our calcium indicator, which is maybe unsurprising because we're directly blocking neural activity, and presumably some metabolic activity is still occurring, right? We haven't killed the cells. But this occurs also, interestingly, across a broader range of frequencies. And we see most attenuation here at low frequency. So we can maybe assume that the low frequency coupling between neural activity and metabolic flux is actually what's causing this attenuation here. So that when we reduce neural activity, that we attenuate the lower frequency component of this signal. Similarly, when we look at the overall network structure, that is, think back to the correlation matrices in the beginning. If we compare the network structure itself before the application, so this is essentially the stability of the network, that is comparing the matrix to itself across time, that it's fairly stable. It's not 100% stable, but that's also a feature of brains. But however, when you compare it to the TTX application, network structure breaks down, and it no longer looks like what it did before application of tetrodotoxin. And this is true for the pyronic network as well as the ATP network. So just blocking tetrodotoxin is enough to block the correspondence between the signals in their regions, which I'll show you as well. But also, like, the more important point, I think, is that it blocks the correspondence across long range correspondences across the brain. That is, that the structure has changed. We no longer have the same structure. And like I said, if we look at it by region, again, you can kind of just ignore the names, other than that the correlation between signals, is mostly non-zero before we apply it to tetrodotoxin, and it becomes mostly zero after the application. If you're wondering, if you're curious as to what I think about why some of these don't go to zero, I'd be happy to talk about that in the post chat, because it is kind of an interesting thing that there are a few regions that don't appear to be affected by tetrodotoxin. And I have some ideas about why, but it's I don't want to get too far off into the weeds about that right now. So clearly neural activity is necessary for the correlation between the two, and also, again, for the structure of the metabolic network itself. So to test sufficiency for this, I express the light-activated channel crimson in a discrete population of cells. So I've moved here now. This is the only part where I'm going to move away from the whole brain imaging method. In a discrete subset of neurons here, this is the antennal lobe, and I'm going to simultaneously activate crimson while I image in this area. And just a quick schematic of what this looks like. So first, I'm actually going to, rather than whole live animal, I'm actually taking the whole head off. And so this is like what you'd call a head capsule, which is kind of a fun way of saying a decapitated fly. And part of the reason I do this is to reduce spontaneous activity, and also just make the fly move less. So it's very difficult when a fly's brain is moving to stimulate a specific region and image it. But what I'm going to do is I'm going to zoom in on the antennal lobes here, and I'm going to look at a specific population of cells called projection neurons. And I'm going to independently activate using a separate laser crimson, and while I'm collecting imaging data from either GCamp or, in this case, the IATP sniffer. And so that's again, questions about this, I'm more than happy to answer about the methodology. And so what I first started, the goal of this initially was to target a very small focal population of neurons and give a very minimalist stimulus. So here I'm giving a very brief pulse of 10 millisecond light that gives me a very modest increase in neural activity. For people who are familiar with GCamp, for GCamp 6S especially, 5% change overall is actually rather small. So I'm trying to get a very minimal stimulation with this. And when I use the same power of stimulus on the ATP sniffer, I also, interestingly enough, see in this case, I suppose that you would say a modest increase in fluorescence to the stimulation. But what's most interesting and most notable about this is that actually how long it decays. And so like I just have here an inlay on the right here showing that the decay for a GCamp signal is rather rapid, whereas the decay to the ATP sniffer is quite slow. And this will come up a little bit in what we think is kind of happening, and why we think that there's a mostly low frequency correlation between the two. And if I increase the stimulation power here to get a stronger signal, I see a bigger GCamp response that also decays a little bit more slowly, because of course these are sort of nonlinear sensors, and calcium itself is a nonlinear response, but also in this case, a huge response to the ATP sniffer that decays very, very slowly. In this case, you can see that it's only back to baseline after several minutes. And so there's a lot of interesting kind of ideas behind this, sort of that essentially that ATP is built, I guess it is built, is produced in response to neural activity, but in a sort of an anticipatory way. And that is in a sort of empty brain, in a dead-ish brain. It's not dead, it's just ex vivo. There isn't a lot of spontaneous activity, but upon producing, very focal activity in just a very specific subset of cells, we can actually see that there's a huge production of ATP, and then that production of ATP persists for a while. And the reason we think this is true in this case is that it's likely sort of anticipatory, and that is that constant neural activity will require more energy, and therefore a bigger pulse of activity, while it produces a sort of, this is about twice as big a response as our 10 millisecond pulse, this doubling of your GCamp response actually produces much more than a doubling of ATP. And that is that because neural activity itself is kind of dominated by low frequency elements itself, that actually like neural activity is predictive of neural activity and therefore should produce more ATP in anticipation. And again, please ask me questions about this. I'd love to talk about it. So that shows those experiments hopefully demonstrate that neural activity itself is required for the correlation between calcium and metabolic flux, and also that neural activity itself is sufficient to drive metabolic flux, and also that it drives metabolic flux in a very persistent manner. But this is, of course, like these are both like either blunt tool or like not very like sort of ethologically relevant, I guess, to a fruit fly, to have its brain covered in tetrodotoxin or to have a single spot on its brain activated while the head is out of it. So we also wanted to look at what a more naturalistic response to this sort of thing would look like. And so into this case, we just are going to use a very simple behavior. In this case, because our fly is mounted, we're going to look at whether or not it's moving or not, right? It's a very binary situation here. The fly is either moving or quiescent. And for whatever reason, flies just kind of alternate between these periods when you have them mounted on a microscope. So it worked out really well for this. So the way we're going to look at analyze this is that I'm back to the whole brain imaging setup, and I'm going to collect either my pyronic or IATP sniffer data simultaneously with gecko data, and I'm going to monitor whether or not the fly is engaged in behavior or not. So here again, simultaneous calcium pyruvate behavior, ATP sniffer as well. And we're going to use basically just going to train a GLM to this, specifically using logistic regression because behavior itself in this case is binary. But basically all this is going to say is that we're going to train this thing to say with our imaging data, what is required to produce the behavior. And then we're going to use that, we're going to apply the model to untrained data and see how well our model predicts behavior in this case. So here would be an example of a model predicting whether or not the animal is supposed to move that corresponds to some degree with it. And then we validate this by just looking at a simple correlation between our model and behavior. So probably unsurprisingly, this works fairly well. Here's a single fly example of what the model predicts will be behavior in blue versus what the animal is actually doing. Again, it's binary in black. And, I mean, surprise, surprise, neural activity is required for an animal to move. We're not reinventing the wheel here, I guess. There is an interesting feature of it, though, that there is structure to this. Again, for the people who are not into fly anatomy, don't worry about it too much. But this is a very specific kind of structured pattern. You can see here in different views of the fly brain. And this does correspond to areas that are sort of essentially considered premotor in the fruit fly brain. Again, not surprising. But what is interesting is that here, this is an example of pyruvate fly. It's nowhere near as good as the calcium imaging data, but we can predict behavior from our metabolic signal to some degree. It's 0.3. It's not terrible. And the weights that our GLM generates across the different brain regions, while it's definitely noisier, also shares some in common structure with the weights that we see our model generate for calcium signals. And of course, because it's noisy, I mean, I'll show you what this looks like once we've looked it over across different animals. And I don't want to spoil too much, but it looks better when you average across animals. So what is that? I'll go through all that right now. So one of the ways that we use to measure this is a receiver operator curve, which basically just means a comparison of whether or not we got a true positive or a false positive in our model prediction. In a perfect model would sort of have this curve be essentially go up to 1 immediately and be flat over here, right? That is, like, you essentially have only true positives and no false positives. And so, like with our calcium signal from JR Gecko, it's a good curve. It's not perfect, but so be it. But interestingly enough, of course, like, if you couldn't predict behavior from your metabolic signal, you would be at unity line here in black. But actually, with our pyronic and ATP sniffer signal, we see above chance positive calls using this model. If we look across this in frequency space, we also see that while it's true that the gecko signal correlates better with behavior across a broad range of frequencies, especially the higher frequencies measured here. For those of you who are maybe like sitting here right now going, why does he keep saying high frequency when this notch right here is 10 to the minus 1? I only mean to say like relative. So for whole brain imaging, we're imaging between 1 to 2 hertz tops. So we don't really truly achieve like a high frequency measure here. Oops, don't want to go too far back. But that it doesn't matter because of course the metabolic signal is so slow that we're definitely capturing the full spectrum required to see that, and that they both drop off at lower frequencies, mostly because behavior itself doesn't, especially when it's weird because it's binarized, but it doesn't have a very large low frequency component to it. But yeah, that basically we see correspondence between behavior and our signals around a sort of middle frequency measured here. And if we look at the sort of peak frequencies and compare the weights again, these are the weights that our GLM uses based on the whole brain to produce these predictions. That again, it's a very nice symmetric, kind of beautiful looking, slightly V-shaped piece here for the fly brain, and that the ATP sniffer corresponds fairly well with it, but it is not perfect, and there are some discrepancies. And that the pyronic also does correspond well with the JR Gecko, or the calcium map, but that it's even sort of more restricted. And if we compare these, essentially the weights across all regions from all flies, it is true that the ATP sniffer mask itself corresponds pretty well with the mask produced for our calcium imaging. And again, like I said, it's not perfect because the metabolic indicators are apparently, clearly not the best way to do this, but they do still correspond well. And the same thing is true for the pyronic mask, where it's lower than the ATP sensor, but it still maintains the overall structure. And so with that, we sort of conclude that movement produces a stereotype pattern of neural activity, and that metabolic signals also capture that. Not all of it, but most of it. So I actually feel like I kind of rushed through that a little bit. I am done with that. I do want to thank very specific people here. So Stefan Denis and Surya Ganguly, who are at Stanford and are in essentially the mathematics department and work with us to build our models and do all of the heavy lifting on that. Tom Clendon, and obviously my advisor, and the other people in our lab who are involved with all the fly work that we do, and Courtney Gallen in the Ghazali lab at UCSF, who does sort of human neuroscience and basically inspired a lot of the work that I did here. So that gives me plenty of time for questions. So I guess maybe rushing works to some degree.
It was a great talk. Thank you. So yeah, if you have any questions, you can either write on the ask a question button down below. You can also write on the chat, and I'll try to pick them up. But yeah, maybe I can. We don't have any yet, so maybe I can start with maybe clarification for me. So in the end, when you were showing the different regions of the brain that would correlate with differently with the different sensors, right? I was just wondering, could it be that maybe, as you were pointing out, specific regions are really sensitive? Yeah, exactly. Maybe specific regions are really sensitive for these metabolic changes, as you were pointing that this was also region specific, the type of structure that you see. And maybe that's why with the sensors,
yeah.
What I mean is maybe there are regions that are regularly required for normal behavior. That's what I mean. So maybe that's why in these you pick higher correlations, and then in the other ones might be required for particular behaviors, which is why more or less in baseline you wouldn't see a lot of structure. Not sure if I'm being clear.
Yeah, no, I get it. I also, realizing now how much I rushed through this, could have added a little bit more here. That what we see here, this shape here basically covers what is known about descending neurons from the fly brain. So, I wanted this to be a little bit more generally understandable without going into too much fly neuroanatomy, but it is definitely clear that this behavior we picked here is just sort of generically recruiting like what we would consider a premotor area. So if you look at the map of descending neurons that have been very carefully mapped by other labs, that the correlation between this sort of like structure here and where descending neurons are the innervation rate, like a sort of amount of innervation from descending neurons, there's a good correlation here. And we actually did test that, and I don't remember off the top of my head what it is, but it's there's a quite a good correlation between the two. And the point, I think, of why, the idea was that we were trying to see if patterned activity would also produce pattern metabolic flux. And the idea behind this is, of course, like, again, this is sort of a very like fMRI sort of inspired idea, where the bold signal itself is essentially, it is a neural activity proxy, but it is also, more importantly, a metabolism proxy, blood flow itself, of course. So, everybody uses that as their neural proxy. And I think here our point was just to be that it's true that even when you use like a fancier sensor with better SNR and like faster kinetics, that we still see blobs that look more or less like the better proxy for neural activity, but that actually it's worse, right? It's like, I'm kind of speaking out of both sides of my mouth here when I say they correspond, but one is better than the other. So like, I don't know, I don't have like a strong way to come down on this other than like, it is true that your metabolic proxy works well, but it is also true that it's slower than neural activity by quite a bit, and that it's noisier.
I see. Well, now we have like 8 questions already, so I think it's good that you guys actually save some time. So we have one already on the chat. I guess it was the first one from Julian Gargulio. So thank you for the nice talk. Would you please expand on the whole brain imaging methods? Is it 2 or 1 photomicroscopy? How do you deal with the fact that Drosophila brain is quite heavily scattering for imaging?
Sure. Oh, yeah, this is all 2 photon imaging. I don't have a so because of the if I just kind of pull up my one of my little schematics here. So when I do this sort of anterior style prep, I'm basically coming at the fly from the antennal lobes back to, I guess, essentially the rear end, the back part of the brain, which is probably the deepest part, is maybe the calyx. And it's only a few hundred microns depth. It's about 180 or to 200. And I don't see too much of an issue with the scattering, right? So I hope that answers that. I mean, it's not too bad. Rita, I can't hear you.
Sorry, I was still muted. Sorry. So yeah, okay. Thank you for the answer. We have another question from Ibrahim. So he asked, very nice talk. He says, very nice talk. And then he asks, do you observe anticorrelated regions for calcium activity that might underlie mutually exclusive behaviors?
Oh, this is a really good question. So in individual animals, you do see anticorrelations. However, a very interesting and robust observation after doing this for years now is that on average, flies, especially like typically when I'm typically looking at resting state or intrinsic correlations, on average there's nothing. If you look at this, there's no blue, right? This is also the case with correlations observed in other animals with calcium imaging as well. So like in zebrafish as well, that negative correlations, at least in sort of resting state, don't really exist or do exist, but are like transient. and that on average, the brain mostly correlates with itself. And so there's a whole other talk behind some of the other work we've been doing here about like measuring essentially what we call a brain landscape, about like what different sort of network structure subtypes the brain explores over time. Is that the bit, and you know, so when we look at that though, it's interesting that the if we do PCA analysis on this, which is like a good first pass, I think that PC1 is always the entire brain. So the entire brain signal dominates everything, which also causes positive correlations to dominate everything. And this again is true in humans. It's true in zebrafish. It's true in worms. You know, it's a weird sort of unknown phenomenon. It's not motion. Yeah, so like there could be, I suppose, there's a graduate student in Tom's lab who's looking at this kind of method, but looking at more intricacies of behavior. They're like, you know, in this case, he's using fly on a ball. So there you have sort of velocities, rotations, etc. And he doesn't do the kind of network correlational analysis, but I'd be curious to ask him if he does actually see in behavior specific ways anticorrelations, because the idea that a behavior suppresses a brain region seems likely to me, but it doesn't pop up in the way that in the analysis that I do, in the way that I collect my data.
Interesting. So yeah, we have another question from Juan Bofi. The question is, imaging implies removing tracheals. How does this would affect aerobic metabolism, ATP?
Well, we have the fly perfused with bubbled saline, and if it's good enough for mammalian tissue, it's definitely going to be good enough for a fly.
Great, thanks. So we have another question by Anita Devineni. The question is, what is the spatial extent of the metabolic signal that's induced by neural activity? European activation experiment probably addressed this carefully, question mark. And then she says, actually, to build on this, I want to ask, do you think the metabolic changes induced by neural activity are cell autonomous? Great question.
Yeah, good question. So this is, we only just started recently doing the activation experiments recently. And one of the first questions we were wondering was how, you know, it takes a long time for it to dissipate in the region where we're looking at, but does it diffuse? And the answer that I have to that is that it does appear to diffuse a small amount, but I also don't, I think one of the things that we want to do in the future in this is like, we looked at one cell population just as a sort of proof of principle for this. And we're looking specifically in dendrites. Well, mostly dendrites of projection neurons. As most of you know, like fly neurons are a little bit more complex than just full-on dendrite and axon. So, you know, I imagine that the kinetics are different in different parts, but it doesn't appear to actually diffuse as much as I would have thought. But I just am not sure like how it applies to other things, and I assume that it's cell autonomous, but I'm not sure if I have actually any way to prove that right now. Anita, we should talk later if you want.
Okay, great. We have another one from Juan Bofi. The question is, metabolic activity seems asymmetrical. One hemisphere only increases ATP sniffer signal in many of your figs with respect to calcium. Care to comment?
Yeah, unfortunately, I think that so like part of the way that we produce this figure is that it's a projection, and so I have the different views of this, like, so hopefully like you can and so basically what it means is that like there's a very, very low weighted region here. I think actually I'm pretty sure this is the mushroom body, and it's true that it's asymmetric here, but it essentially turns this like the projection of this view of it into like nearly zero here, but this like more posterior part of the brain does actually have a more symmetric look to it. I mean, I guess it's impossible to tell what region is what in this like dot based correspondence between the two. Suffice to say that it doesn't look as bad as this thing looks. But you're right that there is some asymmetry here, and I don't know if it's significant or if it's just noise and the way that we visualize it. But obviously you're right. Like even though like, yeah, so for whatever reason, like this guy got a really heavy weight on its like right mushroom body. I don't know.
Yeah, I guess that's what I was trying to hint before. I don't think I was super clear. Like the math, because it's quite symmetrical for our gecko, right? But then for the sensors is well, mainly for the ATP one. It's not super obvious, right? But I guess it makes sense. Like even what you're saying, if you actually go through different stacks, you actually kind of see this symmetry.
It looks better than this, which I realize is not great figure design on our part, but and I actually I almost wish I had Stefan on this call with me, because he's the guy who's like our math pro on this, and we also like realized that like even though there's sort of negative weights, the GLM doesn't necessarily mean that it's like anti-correlated with a behavior per se. It just means that the model just like doesn't use that region, right? Essentially,
it might be sufficient. The other side might be sufficient to explain the behavior, so it doesn't.
Yeah, it's definitely a little bit. I mean, it's definitely a little bit more complicated than I understand enough to get into the weeds about. Other than like, so for example, like in the correlation between the two, like the JR Gecko masks really don't go below 0 with suffice for, except for this sort of one over here. But like the IATP sniffer ones do, but they still like, even though there's a bunch of dots that are below 0 here, they still kind of correlate with the Gecko one, in that like they're more negative when the JR Gecko mask is lower. I mean, this would be a curious thing to talk about if someone is much more mathematically inclined wants to weigh in on it, for sure. This one's a little bit out of my league, though.
Great. We still have like some questions, if you want to. Them. Okay, so we have one from Carlos Ribeiro. He says, "Great talk. Can you comment on possible glia neuron interactions which could underlie the metabolic coupling described?"
Yeah, one of the biggest things that came out of this was what role glia play in, and we suspect a role. I mean, it seems unlikely that glia play no role in this. And fortunately, Tom has a graduate student right now who's been studying glia, and he and I are currently working on methods for looking at. I mean, the question is sort of like, the big question is, can we like monitor different metabolic components, right? So one of the things that I didn't include in this is that there's also a lactate sensor that we have access to, laconic, pyronic laconic. So it's not clear if like the lactate shuttle method is also a thing in fruit flies, although if somebody knows more about this and wants to school me on that, please do. But we're kind of curious to see if we can see metabolic precursors like pyruvate or lactate change in glia in response to neural activity. I've just started collecting data on this right now, and I don't know yet if there's anything there to see, but it is like definitely the next question to ask. Because surely this is like, I mean, I think this is much more well established in mammalian systems, but I mean, I would be surprised if fruit flies don't have something similar.
Yeah,
I think they do have a blood-brain barrier, like.
Yeah. I think the work actually, we've had Stephanie. Oops, we've had Stephanie Schiermeyer, and I'm not sure if she was the one developing that sensor itself, but she has done
maybe, yeah.
some stuff like on glia and lactate and the roles, so that might be cool and maybe related. Okay, so we have still a few more questions. People are really interested, which is great. So we have another one from Chris Rogers, and he says, if you could train the fly to expect a stimulus at a certain time, either through conditioning or predictable repetitive stimulation, do you think you would see a metabolic response that anticipates that stimulus? or does metabolic always lag neural response, as in your Crimson stimulation experiments?
My guess, Chris, is that it requires the neural response. So other labs than us have done pretty good in vitro work to show that calcium and interacting with the mitochondria is what produces the bulk, essentially, like, well, unsurprisingly, mitochondria, powerhouse of the cell. That like calcium is required for.
I'm sorry, Kevin, I'm hearing you in duplicate now. I'm not sure if it's only me again, because I've been having problems with my sound. Could you
Sure.
talk now?
Sure. Yeah. Testing. Yeah. All right. I don't know.
Well, if anyone has issues, just write on the chat. Maybe it's only me.
Okay. Chris says it sounds fine.
It's fine by everyone.
All right. Okay. Yeah. So because like what we know about at least mitochondria in neurons is that essentially they're calcium sensitive. That is that like there's a rapid production of ATP. Well, we think rapid production of ATP. The temporal aspects of this are still kind of unclear, even with our work. That calcium is required for the production of ATP in neurons. So I don't think that there would be necessarily a metabolic anticipatory response. However, there is there is there's like some work that a postdoc I know is doing on like anticipatory behavior in fruit flies, and I'll bug her and see if she is kind of interested in taking a look at that. But yeah, I'm not sure. There might be anticipatory neural activity, which in that case, will produce the metabolic response, anticipation response. Yeah.
Interesting. So we have a couple more questions. So one from Alan K. Have you tried to measure how pH changes with activity in neurons and glia?
Yeah, so this has actually come up from other people before too, and I didn't really think about it too much, but I think my plan was to just actually use just straight up UAS fluorine across the whole brain to see, because of course pH changes. pH is associated with neural activity in that vesicles themselves have a different pH than the surrounding cell, but this might also just be my ignorance, but I'm unfamiliar with the idea that neural activity itself changes the pH of the cell per se. But I guess it might happen in vesicle loading locally enough to change things. I wonder if I guess maybe I'll ask a follow-up question to you. Are you there is some question about whether or not pH itself changes the fluorescence of the sensor, right? So, that's definitely something we're going to check, but I don't think that it's possible for it to change the fluorescence of the sensor on the order of magnitudes that we see. And I also it's not clear to me that it even is in the right direction, the sign of what I would expect to see from either presumably acidification. It's definitely something. I'm definitely going to plan to just take a quick look with just some panneuronal fluorine. It's possible that there are whatever microdomains, nanodomains that do have large changes in pH, but or even modest changes. I think it can't possibly be that large, right?
Yeah. So yeah, the final question is from Samuel Walker, and it's touching on a cool one. So could you talk a bit about the regions where you still see correlations in the presence of TTX? What are your ideas of what underlies these correlations?
Yeah, so if I go back to this, which is probably pretty small. So the two biggest regions that seem to be insensitive to tetrodotoxin are GNG and so the gnathic ganglion and the prow, which are all part of the SEZ, and possibly even the AMMC, which is also, it's not part of the SEZ, but it's also kind of an adjacent area. My thought is that actually that because there's I'm open to a lot of suggestions on this, because these are all regions that have actually quite a bit of input from the VNC. So in a past life, I did a lot of work on the VNC. So like weirdly enough, like kind of know this, but so I think because we're applying tetrodotoxin to the brain, and the ventral nerve cord is essentially allowed to remain in, unperturbed, is that there is actually just like ongoing activity from the VNC to the SEZ. But it's more, in all honesty, was more of a curiosity for us than something we wanted to pursue. But we could possibly go back and look and see like if the neural activity itself in those regions is like robust to tetrodotoxin itself, or if there, it's also possible that we often considered, as like the alternative hypothesis, right? So there's always more than one alternative hypothesis, but in the early days of thinking about whether or not neural activity was the main driver for metabolism, we were of course thinking that obviously a neuron at rest, just like any cell at rest, is metabolically active. So like, we're never going to, completely abolish metabolism in a cell by blocking neural activity because the cell is still alive. But also that, then there's coordination we see across regions. Well, that could potentially be theoretically like coordinated by, you could have like, say, differential expression of something like insulin receptor. Now this is definitely going to be like where I'm out of my league, and Rita can maybe like school me on this a little bit too. But in this world, you could imagine that the bath application, i.e., like hormonal like secretion of insulin, would differentially affect regions, and that metabolism in those cells would be tuned by this, and that would be insensitive to neural activity, and also that metabolism itself could potentially drive neural activity, which I don't think we have fully even ruled out per se. There's a bunch of interesting and older papers that about like sort of overall ion concentrations across membranes and how this is affected by ATP availability. So like in this model, more ATP availability potentially drives more pump activity, which drives larger potential across a membrane, which makes a cell more excitable. We don't have any data to suggest that, but we also haven't really like pursued that per se, like that, but it is on our mind for sure. And like it's possible that these regions, so the SEZ, where the GNG and the PROW are also like postsynaptic to the DILP neurons, like so all of the pars intercerebellum intercerebellar neurons. I'm not going to be able to say that word, I think. So like insulin producing neurons project directly to these regions. So it's possible that there's coordination there, that neural activity itself that is I guess protected from neural activity reduction. So it's a huge question. But my simple hand wavy thing is that, there's postsynaptic to the VNC, but like it could get way more complicated depending on what experiments we want to do next. I think that we're interested in looking at ways to block sort of metabolic output in a way that doesn't completely kill a cell, to see how that affects neural activity. But it's hard to know how to interpret those results, but I also don't have them anyway, so there's no results to interpret yet.
Thank you. I actually think when things get this complex is when they actually get more interesting. So it's kind of a double-edged sword because it's quite difficult to tackle. But I guess it's really cool that you have so many options to explore here. So it's really interesting. So yeah, with this, I would like to thank you for a really great talk and an amazing discussion. I think it was one of the longest and more productive and interesting that we had. So thank you, and everyone who asked questions. Next week we will be having Clemence Bouet will be telling us about the work they're performing. And with this, I would like to thank you all, and maybe we'll take a couple minutes before joining the Zoom so that you have a break. So everyone who's interested, I wrote the Zoom link here on the chat box. So feel free to join us in a couple of minutes. And thank you. See you next week.
Thanks, Rita.